So I think we'll get started. It's a couple minutes after 12 o'clock. So welcome everyone. Today, Dr. Mike and Katie are going to talk about the research grants that we've awarded this year. But first, I'll introduce myself. My name is Eric Wellen. I'm the web developer and technical specialist here at FRAXA. Behind me is my great assistant.
Caleb.
That's Caleb Wellen. He's very excited to be part of this meeting. We also have Holly on. Holly, would you like to give a quick introduction for yourself?
My name is Holly Rose. I'm the Community Services Director here at FRAXA, and I am the mom of Parker, who lives with Fragile X.
Awesome. Thank you, Holly. And just a little bit of housekeeping. If you do have any questions during the presentation, feel free to use the Q&A button down at the bottom. And with that, I'll turn it over to Katie and Mike, and let's go for it.
All right. Well, welcome everybody. We're really excited about the grants that are coming up that are being funded. I'm about to share my screen so we can run through them. There are quite a few. So we will attempt to keep it basically to about 2 minutes per project and leave plenty of time in the hour for questions. And here we are. Okay, so we have about 17, 18 projects, and we've tried to put them in an order that makes sense. I'm kind of the lay translator, if you will, and Mike is the guy with the information. So as we go along, please put questions in chat and we'll handle them, at the end of this whirlwind tour of these great new projects. So first are kind of the curative therapy projects. So the research aimed at curing Fragile X, which we've put a focus on this year. And in fact, FRAXA has a curative therapies fund, which people who want to focus on the cure donate to directly. And this is thanks to them. So starting with at the top, if you will, the Richter Lab at UMass Worcester is working on ASO as a treatment for Fragile X.
And this is a really interesting project because it started out as a study of abnormal splicing events in Fragile X. And what does that mean? Well, whenever you turn a gene into a messenger RNA, that's not the end of the story. A lot of people figure, well, the gene gets turned into mRNA and then the mRNA gets made into protein, but there's lots of different steps in between. And one of those steps is splicing of the mRNA. So the mRNA is made in a particular form and in the original, say, director's cut, but then it's spliced depending on the cell type into different versions of that mRNA before it gets translated, and that makes different isoforms of the protein. Well, Joel Richter's lab had noticed that there were lots of abnormal splicing events in Fragile X cells. And so lots and lots of messenger RNAs from different genes were getting misspliced. And so he wanted to look at that as a way of developing a biomarker for Fragile X. And along the way, what he found was that one of the mRNAs that was misspliced was the FMR1 mRNA, which was really curious because there wasn't supposed to be any mRNA from FMR1 in Fragile X. The gene is supposed to be silenced, but what he found was that it wasn't silenced. In fact, there was lots and lots of mRNA, and it was in two primary forms. One was the normal FMR1 RNA, and one was the abnormally spliced version, which they dubbed FMR1217. But there wasn't any protein, so they still weren't making any FMRP. So that raised a curious paradox. Why do you have some normal messenger RNA from FMR1, but you're not making any protein? So they assumed, well, must have something to do with this abnormal splice variant. So they made what's called an antisense oligonucleotide to block that FMR1 217. And lo and behold, you started to get normal production of protein. So Fragile X cells are now making FMRP where they weren't before. That's really cool. And that has some obvious implications for therapeutics. And so this is true serendipity. They were looking for one thing and they found something else. And as we now know, this technology has been licensed to a major biotech company in the Boston, Cambridge area called Curalis. They were originally founded to cure ALS, amyotrophic lateral sclerosis. But they're very much interested in Fragile X at this point and will be commercializing this. You need to have a company to take this basic discovery and turn it into a product that you can actually put into clinical trials. And before you can do that, you have to test it, do all the toxicology that's required. Curalis they're experts at that. But at this point, you might wonder, well, why are we funding Joel Richter? Because he already made the discovery and now it's in the hands of Curalis and they're pumping millions of dollars into this. Why do we need to fund more work in the Richter lab? Well, they're still working on some of that original biomarker stuff that will hopefully be useful when it comes time to test these ASO treatments in humans. And they're also working on a new biomarker angle because now we really want to know what's going on in CSF, in cerebrospinal fluid, because that's the fluid that bathes the brain. If we want to know what's going on in the brain without actually having to take little chunks of brain tissue out and test it, which is really not a good idea if you ask me, you can look in the CSF. And you might think, well, we could just look for FMRP in the CSF, and that might be one way to see if our ASO therapeutic is reactivating the brain. But it turns out FMRP doesn't get excreted into the CSF. Even normal people with normal levels of FMRP don't have any FMRP in their cerebrospinal fluid. So like when you do a spinal tap and you take out some CSF, there's no FMRP in there. So we can't do FMRP levels in CSF, but they are finding that there's a abnormal expression of other metabolites, things like amino acids, for example, that you see different levels in Fragile X in the CSF. And so they're looking to use that as a readout for the normalization of, for the reactivation of the gene and the normalization of metabolism in those cells. And that's one way we'll be able to tell whether it's really working.
So that'll come around and be handy when Curalis hopefully decides which ASO is the best one, the one that should go to trials, clinical trials, that is.
Because they've actually generated a whole bunch of different ASOs,
Yeah.
and it's going to be important to know which ones are the best.
Exactly. The next one is related. So this company, Quiver, is working with Curalis, which just licensed the Richter discovery. And they're part of this choosing of the best ASO, given their technologies. This is also an interesting new grant for FRAXA because Quiver is a company, not a nonprofit. But we are basically going to fund whatever is needed to get treatments to patients. And at this point, that can include pharma companies like Quiver.
Yeah, what's really cool about the technology that these folks have developed is that they've used this technique called optogenetics. And you can insert a gene, it's actually an insect gene, into any given genome using these kinds of gene editing techniques we've talked about before. It's not exactly CRISPR, but it's the same sort of idea. So you can insert this channel rhodopsin, it's called. And what that does is it flashes light every time the cell is active. Now, you could, if you're studying neurons, poke each neuron with a little tiny electrode, but that's a lot of work. What these guys can do is they can engineer the cells so they flash when they're active. Then they can have a culture of cells which all spontaneously link up together and form little neural networks. And then they can look at how these cells function in networks by looking at these little flashes of light. And of course, you can automate that process so the computer can do that for you and you can get all the data. And so that's a great way of testing new treatment strategies. First of all, you can see, are the cells excitable? Yes, Fragile X cells are very much more excitable than normal. And can you rescue it with particular treatments? And the ASOs are going to be one way to test this reactivation strategy and also to optimize which ASOs. But what's really cool about this is because it's not exactly high throughput, but semi-high throughput, let's call it medium throughput, it means that you can test different cell lines from different people. And so
Yeah.
you can have, we have patient-derived cells, but they're from multiple different patients. And not only that, but we've already talked about how you're engineering so that they flash when they get excited. But you can also engineer them so that you can take a Fragile X cell and you can use CRISPR to fix it so it doesn't have Fragile X anymore. So it's what we call an isogenic control. So that means you've got like the twin that person never had who doesn't have Fragile X, but they're identical in every other way. And so that's something you can only really do in a dish at this point, but it's actually fairly easy to do in a dish. And so you've got these kind of doubly engineered cells. And so you're comparing Fragile X cells with their isogenic controls, and then you can also take normal cells and kind of make them Fragile X using CRISPR and see how those all respond. And you develop this kind of four-way matrix for looking at what the optimal treatment for those cells is.
So
And
this
it's
can really
cool.
be a treatment platform, not just for the ASO project, but also for really any other treatment, gene therapy, et cetera. So,
or just small molecules
or
or drugs.
Yep. Okay. And speaking of gene therapy or gene reactivation, this is one of our favorite teams. We have been funding Dr. Lee's lab, for 5 or 6 years now, and she has discovered ways to reactivate the Fragile X gene that don't involve actually editing the gene itself, which is kind of cool, right?
Yeah, we talk about CRISPR all the time, and it's really exciting to think that you can use this technology called CRISPR-Cas9 to edit the genome. But it's a little more complicated, like everything. And there are a lot of components that go with this CRISPR-Cas9 system. So CRISPR is one enzyme, Cas9 is another enzyme, and then there's a guide RNA that you have to put in there too. So at the bare minimum, you have to put at least 3 different components into the cell and get everything there and assembled at the same place at the same time. That's pretty complicated. But what Dr. Lee found is that if you took an inactive Cas9, which is one of the smaller parts of the system, and you just coupled it with a short guide RNA that would help it to recognize the CGG repeats in the trinucleotide repeat expansion.
The part of the gene the mutation that causes Fragile
Not in the
X.
coding region.
Right.
This is just before the coding region. So you're not messing around with any of the sequence of the actual gene, but just this part of the promoter that has the trinucleotide repeats in it. If you just put in that thing, which is a so-called dead Cas9, it's an inactive, it has no enzymatic activity. It's just a protein that's inactive with a little, very short piece of RNA for recognition of the sequence, which is easy because there's not a lot of these CGG repeats around. If you just put that into the cell, then the cell will take it from there. This binds to the trinucleotide repeat, and the cell does this thing, which is part of the normal DNA repair mechanism, which basically excises lengths of the CGG repeats. And so it gradually, step by step, pares it down, and it takes it all the way from a full mutation to the pre-mutation range spontaneously, all by itself. So that's really, again, sort of serendipitous. I think Dr. Lee was assuming that she was going to have to couple the CRISPR with the Cas9, probably with some kind of demethylating enzyme and various other pieces and parts. So this was just getting started here, but she found that when she just got started, it worked and it kind of fixed the problem. So then at that point, the research has to actually look at, okay, now you do this in cells in a dish. Now can you do this in organoids, in a mass of neurons that's no longer dividing. These are post-mitotic cells. And then can you do it in a live brain? And so there's a number of steps that you have to go through to prove that this is going to work before you can take that into human subjects. But it's all part of the process, and it's all happening now.
So that's where we're at with this grant. This will allow her to do work with organoids because she won a $1 million grant from Harvard Medical School to do the work in live mice. And, we're thrilled about that $1 million that's coming. And that's all needed to bring this to the point where a company will take it forward. So next, we have a new researcher looking at another gene reactivation, gene therapy approach.
So, Hei Young Lee is a researcher in Texas that we've actually been in contact with for a while. She's been doing some very interesting things and has even published some gene therapy studies in Fragile X. And she has proposed a really interesting kind of two-pronged strategy for developing new treatments for Fragile X. Strategy number one is really more of the conventional viral vector gene therapy. But the twist here is that she's going to look at some different brain regions and trying to figure out, is there really an optimal place to try to put the gene in? Because with current technology, you can't really put the gene back in every cell in the whole brain. But if you can find those critical nodes where it's most effective, then you can do it and you can make a real difference, and it'll be persistent. It's kind of a one-shot treatment. And if I had to bet, knowing what we know right now, the one place in the brain is probably the striatum. It's a formation in the very center of the brain that acts as a kind of crossroads for multiple pathways. And so she's going to be comparing insertion of the gene in the striatum versus the hippocampus and looking at the overall output in a live mouse. Like, what does that look like? How much rescue do you get with these two techniques? And then the other one is she's going to use a CRISPR gene editing strategy, the other parallel pathway that'll be done. This is sort of like a double project. It's a 2-in-1. It's got twice the level of funding. And so it's a big project, a big ambitious project. And they're going to try to insert these CRISPR components that we've already talked about before. You have to get all this stuff in. It's really hard to do that with a viral vector. They're not big enough. But one of the interesting new technologies is using little blobs of fat called liposomes. So you're using a lipid envelope as a kind of synthetic vector, and then you're putting the components into that. And in this case, she's going to do it using the mRNA vaccine technology. So instead of putting DNA in there, she's going to put messenger RNAs to synthesize all of the CRISPR components. And so that's a really cool approach that no one's tried before, and it'll be really nice to see if they can actually get this sort of gene editing working. It'll be the same progression in cells in a dish, organoids, and then mice. And in the case of the mice, you can at least do a first pass kind of test with the knock-in mouse for Fragile X, which is not a model for Fragile X at all. But if you knock in the CGG repeats, you get a mouse that has 300 or 400 repeats, but it doesn't silence the gene. But that's a good model for studying FAXTAS. But in this case, if we can pare down those repeats, we'll be demonstrating some effectiveness. And that's really exciting.
Wanted to take a minute to say, a little bit about organoids, because the next several projects are organoid based, and some of the last ones you've seen already are too. So, most Fragile X research has been done on mice, but in the mouse that's modeling Fragile X, they just knocked the gene out altogether. So there's no way to test repairing the gene because the gene is not in that mouse. So what do? One really cool approach is organoids. So these little tiny pinhead, three-dimensional balls of cells start from a cell from a person, say your son, my son, and it's from blood cells. So you take a blood draw and they reprogram blood cells back to induce pluripotent stem cells and then grow an organoid from that. And there you have this little sphere, which actually builds connections in different brain cell types and behaves in many ways like a little brain. And so that is a great test bed for drug treatments, gene therapy treatments, ASOs, anything really. And the other thing to think about is how personalized this could be. So an organoid from Andy's blood is going to look different from an organoid from somebody else. So it's become a real breakthrough for the field. And you will see that with the next few, such as this.
So Nicola Elvasore is in Italy. He's in Padua, and he's been doing some work on Fragile X that's been funded by FRAXA for a couple of years now. They've been looking at a lot of the mechanisms of the silencing of the gene and looking at differences in methylation patterns and how that affects expression of different genes. In this case, they're going to convert over to looking at organoids. And the twist here is that they're not only going to be looking at these organoids, these little brain balls in a dish, but they're also going to compare some of those results with these human cells that have now been grown in a dish and then transplanted into mouse brain. So you can actually take these organoids whole and transplant them into a mouse brain, and they will become incorporated into the mouse brain. And they'll still retain their internal structure, but they'll also make connections with the mouse brain cells around it, and they'll get vascularized. And normally, induced pluripotent stem cells, any kind of stem cell work, is very labor intensive because you have to keep these things alive. It's like having plants that you need to water 4 times a day. It's really hard keeping these things going. It's very labor intensive. But in this case, if you transplant it into the mouse, then that can actually persist in there for a long, long time. And so they get a chance to study that over a very, very long period of time and look at that. Really interesting and exciting prospect, I think. And Peng Jin and Zhijing Wen, it's really hard for me to pronounce his name, but it's probably even harder for him to pronounce my name. But they've been working on organoids at Emory for quite a while, and they're actually interested in preclinical testing of various therapeutic strategies, including small molecules in organoids, and they have lots of human stem cell lines that they've developed over time. They probably have the largest repository anywhere. And so they're very, very good at working with stem cells. They're really good at working with organoids. They've done a lot of biochemical and electrophysiologic testing on these organoids. And now they're going to look at a number of different drug classes, a particular focus on the phosphodiesterase inhibitors.
Such as Zatomalast, which is Shionogi's drug in trials right now.
But many more good PDE inhibitors to come, as you'll probably hear over the next couple of years, especially if this is successful. So they're just really, really good at working with organoids and very experienced in Fragile X. And they're interested in doing things that are more and more preclinical, and we are supporting that.
Next.
This is a new lab for us, although they're at the University of Cincinnati, right down the hall from Craig Erickson and his lab. And they're going to be looking at organoids. And a lot of these are from patients at the University of Cincinnati who are well characterized. Once again, we talked before about how you can take cells from a patient and make these organoids. And then the question is, how representative of that patient's condition is this organoid? Well, first of all, you have to have a well-characterized patient. And Dr. Erickson, of course, has patients who are not just clinically well-characterized, but they've all had electrophysiology studies done on them. They've all had EEGs done. They've participated in research protocols, sometimes in drug trials. And so you've got this clinical history of the whole human being, and now you've got their cells in a dish. Now, in this particular case, there's a focus to the project, because most of these projects do have a specific focus, even though they have something that's more generalizable about the project. And in this case, they're looking at a particular pathway that's involved in methylation of DNA. So they're looking at, or excuse me, methylation of messenger RNA, and looking at ways to modify that with drug treatment, and then to observe those changes in the organoids. So this is
Really
both an
cool.
organoids project in general, but also specifically working on a new target for treatment and potentially a new therapeutic strategy, a new drug class.
One thing that's interesting to me, a lot of people, including me, think, okay, these are the curative therapies and these are the drugs, the targets. This is a drug, but it's targeting the middle, not the DNA, not the gene, but the RNA. And so when you think about how much a drug could do, if it's something like this, it might do quite a lot, just like the ASO targets the RNA, the middle step. It's something to keep in mind. It's a whole
And I think
really.
also the methylation of these messenger RNAs is also connected to the splicing of the mRNAs. That's part of the tagging process that occurs. And so you can see how these things are all connected at different levels. So this is not entirely unrelated from what Joel Richter is doing, but everybody has a little bit of a different focus on a different part of the process.
Now, this project is co-funded with the Autism Science Foundation, who we work with closely and have for quite some time. And so this is both Fragile X focused and autism in general focused.
And what's interesting here is that Maureen Krish has previously worked with Rudy Yanish at MIT and looking at reactivation strategies in Fragile X. And along the way, she became expert in this technology for engineering these hybrid animals. So you take a mouse, it's a normal mouse, but then you're able to put some Fragile X human cells into that mouse brain. And we referred to this previously with the Elvisori lab and putting in the organoids. In this case, her specific focus in this particular project is on the microglia, which are one of the glial cells, the non-neuronal cells in the brain, and looking at the influence of microglia on the Fragile X phenotype by taking Fragile X patient-derived microglia and putting them into a non-Fragile X mouse to see how that affects the mouse's behavior. And you can also go, obviously, later on and dissect it and look at different aspects of the fine structure of the nervous system as well. And this is potentially a very important way of studying not just a number of cell types in Fragile X, but also a number of different conditions. Because you can imagine, you might have somebody with idiopathic autism. They don't have a genetic diagnosis. They probably have something going on genetically, but it hasn't been diagnosed. But you can take stem cells from that patient and make microglia out of them and put them into the mouse and look for the differences in behavior in that mouse afterwards and how those microglia are functioning in that mouse-based system. So it's a really potent model for studying a number of different neurodevelopmental disorders. And that's why we're co-funding it together.
Next, the live mouse tracker in Belgium. And we've been funding this for several years, but now they've really got it optimized. So they have this platform where mice live together and play together in a kind of a normal group environment, but they are tracked. Their behavior, their activity, everything is tracked in great detail.
Yeah, and a lot of the studies that we do preclinically, we'll put the mice into a cage, and there'll be a little video camera there, and there'll be some computer tracking software that tracks the mouse around. And that's good. That's great for one mouse if you're just looking at kind of two-dimensional tracking around a cage. But what this does is you put a little RF chip into each mouse, and each mouse is unique and distinguishable from all the other mice. And then you have sensors around the cage, and you can put in 2, 3, 4, 5, 6, however many mice you want. and you're not requiring them to do anything. You're just observing them interacting, and the computer takes care of it all, and it tracks how they move around and how they interact with one another, and it generates a vast amount of data. And when you do this, you see a very distinct difference between the Fragile X mice and their wild type litter mates. And then if you treat these mice with things that we think might be therapeutic and some things that we know are therapeutic, you see that that profile of behavior actually corrects. And so it's a really powerful way of testing drugs, and it looks at a broad spectrum of behaviors kind of agnostically. You don't really have to have a theory about why this drug is working or what it's working. It just tells you that it makes the behavior of this mouse much more like the wild type behavior in any number of different ways. And so it's really powerful for preclinical validation, especially going forward, as we're going to have more and more companies, hopefully, that want to validate compounds before taking them to the clinic. So really cool stuff.
Yeah, so just a moment to mention our, until this came live, if you will, the one facility that FRAXA supports to test, to validate treatments is down in Chile, and hundreds and hundreds of compounds have been tested there. And we kind of feel like that's been the most important single project. And so we need more like that. And this is one. We need more objective ways to test possible treatments. Next.
So we still have a number of projects that are aimed at helping with the development of specific drug classes. You know, we've got a big, long pipeline of things that we've shown can be helpful in Fragile X, and now we just need to do clinical trials. But there are some classes that are particularly interesting and promising. And one of them is it's been shown that bumetanide is a drug that controls intracellular chloride concentrations. It's marketed as a diuretic, and it's a very potent diuretic. But you can also manipulate chloride levels by using bumetanide. And in the Fragile X animal models, what you can show is that there's this switch, and that a lot of inhibitory cells early in development are actually excitatory, and they're relying on a chloride switch to turn them into inhibitory neurons. And in Fragile X, that just never happens, or it happens much later in development. So the bumetanide flips that switch and converts them into the normal, appropriate inhibitory neurons. The problem with bumetanide is it doesn't get into the brain very well. And in clinical trials that have been done so far, it's impossible to have a placebo control, because when you take bumetanide, you just start peeing like crazy. So everybody knows you're on the drug. There's no more placebo control. And that's confounded a lot of the clinical trials. So this company, IAMA in Italy, has developed an enhanced version of bumetanide. They've tweaked the molecule, and they've got it so that it's brain penetrant, and it's not really a diuretic anymore. And so they've already done some initial testing in a number of autism model systems that have shown very promising results. And this is a project to basically do the validation of this compound in Fragile X model systems, and it's IND enabling. So
Yeah.
Investigational new drug will go into clinical trials as a result of this.
And this is a case where Laura Conchetta is a co-founder of IAMA. So she's already got the corporate structure to take this toward the clinic. So that's kind of nice.
And Kim Huber, of course, is legendary in the Fragile X field. She's someone who's been one of the most productive researchers in the field, worked for many years with Mark Baer, and now she's working very hard with Craig Erickson in this large center to look at electrophysiology in various model systems of basically cross-platform analysis of electrophysiology in Fragile X. And in this case, she's looking at sex differences and the role of estrogen receptors in especially inhibitory circuits in Fragile X. And one of the interesting things about Fragile X is, you know, we think of males with Fragile X who are very similar to the knockout mice, in that they don't really have any FMRP. But females with Fragile X, well, they still have one good X, so they're not anything like the knockouts. because in the mouse model, you can actually have a double knockout. You can have a female with both copies of the gene on both X chromosomes knocked out, and they don't produce any FMRP. And you might think they'd be pretty much just like the males with the knockout of the Fragile X gene. But they're not. They're actually not at all like them. They differ in a number of significant ways. And so that's an important model for studying sex differences in autism spectrum disorders. And that's basically the heart of what she's doing, is looking at these specific mechanisms and how it involves estrogen receptors in this case.
We are trying to put a focus on research on females, since, girls with Fragile X really are very different in general than boys with Fragile X. And, it's important to understand that, because the treatments may be very different. Okay, just a few more, and we'll
Just
get to
quickly,
questions.
Ben Auerbach is another a graduate, if you will, of the Bayer lab at MIT, and now he has his own lab at the University of Illinois. He's working on the Fragile X rat model and looking at the regulation of protein synthesis and how that's altered in response to auditory stimuli. That's something that's been worked on a lot in the Fragile X mouse, but it hasn't been studied as much in the rat. And there may be some differences there, and there may be some advantages to the rat system that will allow for some new insights into these mechanisms. And it's been a very productive line of research in the mouse model, and I suspect it will be in the rats as well.
Rats are smarter than mice, just
And
FYI.
bigger. They're bigger.
And bigger.
There's a lot more stuff there to work on. They're much bigger.
Bigger brain.
And just quickly, these are folks that work with Peter Kind at the University of Edinburgh, the Patrick Wild Center. So there's a lot of Fragile X and autism research going on there. I alluded to work on microglia, which are one of the major glial cell types. The major cell types in glia are astrocytes. Those are the ones that work in the gray matter of the brain to set up the environment for the neurons. The microglia are kind of like the immune cells of the brain. And then the oligodendrocytes are the cells that create the myelin sheath. So when we think about the white matter of the brain, the white matter is the fat, which is the myelin, which is the oligodendrocytes. That has not received a whole lot of study, even though some of the imaging studies in Fragile X have shown that there are some white matter abnormalities. These are kind of the long axons that go from one part of the brain to another. So it's not as involved in the kind of local processing, but in the coordination between different parts of the brain. And it turns out, unfortunately, like most things, when we look closely enough, we find some abnormalities there. And there are some abnormalities in oligodendrocytes in Fragile X, and it hasn't been studied very well. But there are a number of things that you can do. There are a number of different interventions for white matter abnormalities, and there are a lot of other, there's a lot of development in this area in pharma companies, and so there's a lot of potential there for potential treatments for this, including things like clemistine, which is just an antihistamine, which is commonly used as a way of enhancing white matter function. So really important and overlooked area, kind of basic, but could very well turn out to be productive. Another thing that's been relatively overlooked, but which is clearly a problem in Fragile X, is mitochondrial function. And there's clearly mitochondrial dysfunction in Fragile X. And whenever you're looking at metabolic abnormalities, we're talking about the energy production and utilization inside the cell, just on a single cell level. And the mitochondria are kind of the powerhouse of the cell. Or I think of it as like the boiler room. This is the boiler room that keeps your locomotive going. And the mitochondria are in charge of actually producing the ATP. That's kind of this universal energy source for the cell. And it's clear that in Fragile X, we have excessive energy demand because the brains of kids with Fragile X are kind of hyperactive. They're a little bit too big and a little bit too active. They don't have the inhibitory tone to keep things in check. So there's a lot of demands metabolically, but at the same time, the ability to produce energy seems to be in some subtle ways corrupted. And that's exactly what they're going to be looking at here, is how to restore that balance so that you can get a situation where you're not always running in the red zone. I always think of it as like, You're out there on your bike and you're riding as hard as you can. When I was a little kid, I used to watch the Tour de France and I would wonder, well, why doesn't that guy in second place just go faster? Just pedal faster. And he's already pedaling as fast as he can. That's why. And so you're already in the red zone. You're already operating at your maximum capacity. And in Fragile X, I think our kids are spending a large part of every day in that red zone. And the idea is to try to bring them down to a level one way or another on one side of the equation or another where they've got a little bit of extra capacity.
That resonates.
And this one is another one that's a little bit basic, but we have some new technology that allows us to actually look in real time at how the gene FMR1 is operating, how it's being utilized in the brain, how it's being transcribed, and how the kinds of abnormalities that we see when it's not there, when you have no FMRP and the gene is not functioning properly. How are you unable to regulate gene expression from one part of the brain and one cell type in the brain to another, because it's different in every cell everywhere you look? And up till now, the way we assess this stuff is we just take the whole brain and we put it in a blender and we do an analysis on it.
A mouse brain. You're right.
Yeah, mouse brain, just to be clear. And you're not really looking at different parts of the brain. You're just taking the averages overall. But that's not how any of this works. It all works from one cell type in one region to another. And so this is going to actually try to look at that using some very advanced technology. Fairly basic, but will almost certainly generate some really major insights.
And Oswald Stewart is a real giant in the field. So they're the good ones.
He hasn't gotten the Nobel Prize yet. But
not yet.
Will.
Not yet. It's coming. And last
but not least,
least,
this gets to the metabolic angle again. And Tom Jongens was actually the first person to ever suggest that there were metabolic abnormalities in Fragile X through his work in the fruit fly model. And the reason for that is that fruit flies live a kind of fast and furious life. And it's pretty easy to see if there's a metabolic abnormality in fruit flies because they're dying all the time. And you get to see them just die a little bit more quickly in Fragile X. And that's exactly what they observed. And he's been studying insulin signaling and the crosstalk between that and cyclic AMP pathways in Fragile X. And Tom was actually one of the first people to publish on cyclic AMP abnormalities and the use of phosphodiesterase inhibitors as well. So, he's got a long history of productive Fragile X research, and they're really drilling down into these mitochondrial abnormalities and how those influence these changes that we're observing in metabolism.
Yeah. And there you have it. I'm going to, just a thank you slide, and also where you can go for continued news. All of these slides you saw will be posted on the website over the coming months with more details. At the bottom is the science section if you want to delve into them. As I say, they'll be coming as these grants ramp up. And if you want to subscribe, you'll get notices of them, or you can always check fraxa.org/news and you'll see the latest updates there as well. So with that.
Oh, wow.
Thank you.
Wow.
Thank you, Caleb.
Caleb thinks you guys did great. So we haven't really had any questions come in yet. So just a
reminder. Really? I see.
So just a reminder, if there are any questions, use the Q&A button at the bottom, or you could also raise your hand right now. And we actually do have somebody raising their hand. All right. We will allow you to talk. You should be able to ask your question once you unmute.
Hello?
Yes.
Hi, we can hear.
Hi. Everything sounds so groundbreaking and wonderful, but I'm just so afraid about will any of this actually come to the surface, like not in our lifetime? It's just really scaring me that how long everything is taking, and it just won't be in my lifetime, and I'll just never see any of this really come in effect for my son.
Well, the first thing you can expect is to learn this fall if the trial of Zatomalas, the 2 trials, are positive. Because if they are, that will be coming in your lifetime. And in fact, in the next year. So that's the short answer. As far as the other ones, they follow. I mean, it's really a big junction point. If that's positive, then things go fast track. If it's negative, we'll be very disappointed, but there is another company that's right on the heels with a very powerful drug, and they're getting ready for trials. But overall, how can we tell? We don't know. We are obviously doing everything possible with the funds that FRAXA has to bring this stuff forward.
Yeah, so they've completed enrollment for the Zynerba, that's the CBD trial, and for the Shionogi
Yeah.
Tetra PD4D trial. So if either of those is successful, I think we'll see a real explosion in the field. If not, I think a lot of people see a real dichotomy between these curative therapies that are just restoring Fragile X protein, FMRP, versus small molecule therapies. And even if everybody says, well, small molecule therapies it's just too complicated, no small molecule is going to fix Fragile X. Well, the companies that are working on these more curative therapies, if anything, I think they'll be encouraged by that because they won't have as much competition. Believe it or not, people worry about stuff like this in the biotech industry. I don't think it would be a problem if there were one or two drugs on the market for Fragile X. I think there'd still be plenty of room for improvement. But some of the investors will say, well, it's getting to be a crowded market, and they'd be happy if they saw a trial failure from Zynerba or from Tetra and Shionogi. I would not be. I would like them all to succeed. But I think the Curalis timeline for the ASO therapy is for clinical trials in probably realistically about 2 years. But I think you're right, Diane. Everything takes way longer than we would like it to take. It takes way longer than you think it should take. But that's true all the way up and down the line. The science is the same way, even just the basic science projects. And you get this awesome result, and you're thinking, oh, when is it going to get published? And then it turns out it takes 5 years for it to just get published.
Yep.
It's like, why? Why does it take that long? It's like every single thing just takes way longer than you think it should. And that's part of what we do, is to try to accelerate those things and to try to relieve those bottlenecks. But there's only so much we can do because there's only so much money we have, and there's only so fast that you can push the science.
And the regulations, all the regulations out there to keep everybody safe, they all take an incredible amount of time. It's quite an education. So
But
we're in the weeds.
Zatolmilast could be happening as an available drug in the spring. Yeah, so fingers crossed.
Excellent. You had mentioned quickly there, you were talking about like the ASO, and there was a question from Heather that said when ASO breakthroughs made the news, it was expected to be about 5 years from use as a therapy, I think is what they said. Do you feel that's still true? Like, what are your thoughts on that progression?
Well, I'm hoping it'll be significantly less than 5 years till we have clinical trials. One of the nice things about ASOs is that it's very modular. ASOs are these kind of plug and play things. You change the sequence of the oligonucleotide, and that's the only thing that really changes. Your overall product remains the same. The way you do the clinical trials remains the same, but you still have to do the toxicology on your product. You still have to optimize your product, do the toxicology, and then go into clinical trials. But it's fairly quick compared to small molecule development, actually, and that's one of the appealing things about it.
Then we also had a question from David. He said, hey, Katie and Mike, great to see you again. Quick question about the ASOs. Any thought on administering these? I guess they can't be given orally easily as they'll get broken down. IV possible, I guess, but will they penetrate the brain? Possibly injection directly into the CSF?
Unfortunately, that's the scary part, is that basically it's like doing spinal anesthesia. You're administering it by a shot into the spine. not into the actual spinal cord, but into the CSF surrounding the spine. And that's why we're talking about CSF biomarkers, because while you're in there, you can just pull some of the CSF out and you can use that to look for your biomarkers. But you administer it by an injection, basically like a lumbar puncture, like a spinal tap. Now, that sounds kind of barbaric and kind of crazy that anybody would do that, but they're actually doing this in Angelman syndrome. And Liz Berry-Kravis is doing 2 ASO trials for Angelman syndrome right now. And she's reporting that people tolerate it really well. They keep coming back. They're not having major problems. Sometimes people will have a headache the day of the procedure, and that's usually the extent of it. So it's been very well tolerated, and people are doing it, and they're seeing people responding to it as well. So it sounds awful. In practice, it's not so awful. And you don't have to do it very often. So we're talking about once every 2 or 3 months.
Oh no.
Excellent.
Oh no.
No.
Oh,
Yeah,
Caleb's getting a headache just thinking about it.
Caleb had some amazing commentary. I'm glad he was on mute because he was presenting their document. Next up was there was a question here. Are there any connections to German companies or organizations?
You know, one of the great things about what FRAXA does, and the most important part of my job, is that we work with a bunch of different pharma partners on a bunch of projects. And so we have about 30 different active pharma partners at any given time. And so I'd really like to tell you about all the great stuff that we're doing with them, but that's all the stuff I can't talk about. Because I mean, there's a certain point at which I will be able to talk about it, and then you can't shut me up. But until then, it's all pretty confidential. And so I can tell you, yeah, there are a couple of different German companies that we've been working with, if anybody's interested specifically in Germany. A lot of times these days, you know, things operate internationally across borders. It doesn't necessarily matter what country the company is in. But we're working with a couple of Spanish companies. We're working on a number of different French companies, German companies, several companies in the UK, a couple of Canadian companies, a couple of Australian companies, and then a whole bunch of other
Japan.
companies that are in the, oh, that's right, a few countries. It's a bunch of
Quite a
companies in
few
Asia now.
yeah.
That, since Shionogi got involved, now everybody in Japan wants to get involved as well.
Also, we're funding a team in Germany. We didn't have time to run through the ones that are, you know, got started last year and are being renewed, but it's a cool project. And they're on the website.
Yeah, and it's related to the insulin signaling abnormalities in Fragile X, and they've gotten some really cool results already.
Yeah.
It's already been renewed for a second year. So
Yeah. Just today, actually.
All right. Next up was can you clarify a little on Zatomalast? Like, is the study closed? What's like the current state of it? Really, there's been a lot of questions about that. So maybe just a bit more.
Yeah, I can. The study is fully enrolled, so closed. It's a 13-week study, and it's already in the last 13 weeks. So when I say it, there are really 2. There's the adult study and the adolescent study. The adult study is going to finish first because it's well into that 13-week, window. They have to wait till the last patient finishes the 13 weeks. And the adolescent trial will be, a month or 2 later. So we're talking about, learning something in the fall. What we don't know is, after the data's opened, how complicated it's going to be. So it's not like they can open it on Tuesday and tell us all on Wednesday. It's probably a couple of months of poring over the data and so forth, but figure on the fall.
yeah, and it should be interesting because they'll have all of the data from the randomized controlled trial, this placebo-controlled double-blind phase, but then all the data from the open-label extension as well. And generally speaking, the protocol mandates that you don't start looking at anything until everybody's done. So if you're wondering why they haven't, done all the ones that are finished already, you
They
can't do
can't,
that.
they can't
It's not
do it.
allowed, not allowed.
FDA
They'll
rules.
bias the results.
Yeah, wish they could.
And
They
just,
have no idea either. Go ahead.
I was just going to say, and just to put some comfort in people's minds, those that are on the trial, can you elaborate a little bit on the open label, like it's continuing?
Yes, just because this is all happening doesn't mean that people are going to lose access. So if people are in the continuation and are taking the drug now, they get to keep taking it, which is great.
Excellent. All right. Next up, we had a question from Diane. It was, can you share like some of the Canadian companies that we work with?
No.
I assume that was the answer. But
I would love to talk about all these great companies that we're working with, all
You
these
could
incredible ideas that people have, and all
You
these
could go to the
fantastic drugs.
website and search on Canada and see the grants that we funded. And you will see mention of ADMARE, ADMARE there, because what the team up there is working with them. So that's public,
And
but
if
that's
I could
about
make
it.
a suggestion, if anybody wants to dip their toe into the AI revolution, you could go to Grok or ChatGPT, just free versions, and
And
just
ask.
type in, what companies have publicly expressed interest in developing drugs for Fragile X.
That's a
And
good
it'll
point.
give you a big long list. The only warning is that I see press releases every day from companies that are saying that they're developing something for Fragile X that I've never heard of. They've never contacted us. They're not doing anything relevant as far as I can tell. So there's a little bit of window dressing that happens in some companies where they're trying to raise money. And if maybe one indication is considered to be kind of trendy these days, they'll just throw that out there. And they'll even show on their website that they've got like some phase 1 pending or something like, and it's kind of, in some cases, it's complete nonsense. But it will give you a list of some of the companies, and most of our partners will be on there. But I can just tell you, there's a few really good ones that won't be, because some
They're not
companies just
ready.
they're just really secretive about this stuff. They don't want their competitors to know what they're up to, and yeah.
But we're glad they're in the mix. Any one of them could be key.
all right, that wraps up all the questions. If there's any more, you guys can throw them in the Q&A. We're pretty much done with the presentation. Just a reminder, this was recorded, and we will be publishing it to YouTube once we get that done. We'll send out an announcement on our newsletter, so be sure to subscribe to that. There'll be a link in chat where you can do that. You can also subscribe to our YouTube channel if you want. You'll get a notice when that pops up there. That is also in chat, and we did get a message from Dawn, so we'll definitely take that one. Dawn said, I want to reach out to mom who asked how long till the treatments?
You want to connect?
Yeah.
She wants to
Yeah.
connect.
No, she just wants to give a little bit of insight here from her perspective. So she wanted to reach out to the mom who asked how long it'll take for treatments and will that be in our lifetime? And she wants to say, having known FRAXA for approximately 30 years now, they have come a long way since the beginning with fruit flies. All of these studies highlighted today are so exciting and promising. As I hang from my own cliff on a daily basis, FRAXA gives me a lifeline of hope for a better future. I have many other projects I want to work on, but Fragile X is my first priority. So, nice
Wow.
words.
Oh,
Thank you.
That's very
couldn't have
sweet.
said it better
Thank you.
myself. Thanks, Don.
Yeah. Yeah. All right. Well, thank you, everybody. And
Fiona?
stay tuned, I guess. Yep.
Wait.
Anybody wants to get involved in World Day, just contact Holly, and it's going to be exciting.
It's looking good, coming up fast.
Yeah. Yeah.
All right, thank you, everyone.
Bye.
Bye, everyone. Bye, Caleb.
Bye.
Bye, Caleb.
It's right. Oh my God, it's right.